Hi, it's Dr. Phoenix. Today we're going to be learning about meiosis. We've just finished looking at mitosis, and we've seen in mitosis that cells undergo nuclear division in such a way that they create an exact duplicate of the genetic information from the parent cell into the daughter cells. So the number of chromosomes that each daughter cell receives is exactly the same as the number of chromosomes that the parent cells had. Now, in meiosis, we're going to see what's known as a reduction division. In organisms that reproduce sexually, the chromosome number has to be reduced in half so that when for instance, sperm and egg unite, we're not resulting in a doubling of the chromosome number. So for instance, in humans who have a total of 46 chromosomes in each of their body cells, the sex cell chromosome number has to be half of that so that when the sperm and the egg unite, 23 plus 23 in each of the sperm and the egg will result in a new cell that has 46 chromosomes. So meiosis is the process of basically forming gametes. Gametes are sex cells, and meiosis results in the chromosome number being reduced in half. Now, all organisms have different numbers of chromosomes. As I mentioned, humans have 46. A lot of studies are done on fruit flies because they have a short lifespan and only 8 chromosomes, so they're a little bit easier to study. So fruit flies having 8 chromosomes total, 4 came from the male and 4 came from the female parent, for a total of 8. So when we look at these chromosomes in a fruit fly, we're going to see that they're in pairs. One came from mom, one came from dad, one from mom, one from dad, one from mom, one from dad, one from mom, one from dad, and so on. We would describe these pairs of chromosomes as homologous. Homologous chromosomes are identical chromosomes in terms of they carry the genes for the same traits. So if this chromosome came from mom and this chromosome came from dad, and they're carrying genes for eye color and this particular enzyme and this particular enzyme, then they are homologous chromosomes. The genes may not be exactly for the same trait. For instance, mom may have a gene for blue eye color and dad may have a gene for brown eye color. But regardless, they are carrying genes for eye color. All right, so these are homologous chromosomes. They are identical pairs. And so in the fruit fly, the green chromosomes would be a homologous pair, and the orange would be a homologous pair, and the red and the purple would be homologous pairs. They are all, each pair are carrying genes for the same traits. And one of those pairs came from mom and the other pair came from dad. Now, if a cell contains homologous pairs of chromosomes, we call that cell a diploid cell. A diploid cell, meaning it has homologous pairs, basically. And we represent the diploid number in a cell by using the symbol 2n. 2n. So in fruit flies, the diploid number is 8, which can be written as 2n equals 8. 2n means the diploid number. And you'll see what the n means in just a second. 2n equals 8, or the diploid number equals 8. The sex cells, as we just mentioned, have half those numbers of chromosomes. And we refer to that number as the haploid number. All right, if you reduce the chromosome number in half, that's referred to as the haploid number. And that is represented by the symbol N. So the haploid number is represented by the symbol N. In this case, the haploid number is 4. N equals 4. So you can see now that 2N would equal 8, and that would be the diploid number. Alright, so in order for gametes to be formed, the diploid number has to be reduced to the haploid number, and that process is referred to as meiosis. Meiosis is the process that separates the two sets of homologous chromosomes so that each gamete ends up with just one of those chromosomes. So here's our diploid cell. These two have to be separated. These two have to be separated to become a haploid cell. And this sex cell, or this gamete, now has the haploid number. And this is what we're going to be looking at here in the next few minutes, how a cell produces the haploid number from the diploid number. And this process that we're going to be looking at is called meiosis, of course. So the definition for meiosis is the process of reduction division in which the number of chromosomes per cell is cut in half through the separation of homologous chromosomes in a diploid cell. Now, meiosis involves two divisions. In mitosis, we just saw one division that started with prophase, ended with telophase and cytokinesis. We're going to see that that has to happen twice, meiosis 1 and meiosis 2. One of those is quite a bit different than mitosis, and the other one is just like mitosis. But the end result is that the diploid cell that we started with is going to be is going to become 4 haploid gamete cells. Now, before we start, I want to refresh your mind, because this is where the big difference is between mitosis and meiosis. If you remember, during metaphase of mitosis, the chromosomes all, underline all, line up in the middle, and then the sister chromatids will separate in anaphase. All right, this is the big difference that we're going to see between mitosis and meiosis. So just remember this getting started. In meiosis, we're going to see during metaphase 1, during the first part of meiosis, that instead of all the chromosomes lining up in the middle, they're going to line up in pairs like this. Right? And that's going to be the big difference. So let's continue. All right, so first we're going to start meiosis by going through a phase of interphase where, just like we saw before, the chromosomes are going to be replicated or duplicated, and the cell will be getting ready to undergo some sort of cell division, of course. Then as it enters prophase 1, just like we saw before, the chromosomes are going to start to become visible. The nuclear membranes are going to disappear. The spindle fibers are going to start forming. That's all the same. But what's different is the homologous chromosomes are going to find each other. So chromosome pair number 1 will find each other, and chromosome pair number 2 will find each other, and so on. And they're going to form what's known as a tetrad. They're going to line up side by side, and they're going to form a tetrad. Tetrad means 4, so there are 4 sister chromatids if you count them in a tetrad. So that's what happens in prophase 1. Now, during this time, when the homologous chromosomes find each other, many times parts of the sister chromatids will break off and they'll switch places. And so there'll be some genes that will actually be switched over from one chromosome to another. And this is called crossing over, which is an exchange of portions of their chromatids during this prophase 1 stage. All right, now remember in metaphase of mitosis, all the chromosomes lined up in the middle. Here's the big difference. During metaphase 1 of meiosis 1, look how the chromosomes line up in the middle. Instead of all of them lining up in the middle, they line up in pairs. So that when anaphase begins, now what we're doing is separating homologous pairs. We're separating the mom and dad chromosomes rather than the sister chromatids as we saw before. So what ends up happening now, by the end of telophase 1, the chromosome number is going to be reduced from the diploid number to the haploid number at the end of meiosis 1. We started with 4 chromosomes in each cell. Now we're ending up with just 2 because we've separated the homologous pairs. Now as we enter meiosis 2, we're going to see that the daughter cells that came out of meiosis 1 will go straight into prophase 2. There will be no interphase. There'll be no more replication of the DNA. That's already happened. And so from meiosis 1, they're going to jump straight into prophase 2 of meiosis 2. The steps of meiosis 2 are basically identical to mitosis. So in prophase, again, we're going to see the nuclear membrane disappear, spindle fibers forming, the chromosomes are going to be visible. Now during metaphase 2, now we can see all the chromosomes are lined up in the middle just like we've seen before. During anaphase 2, the sister chromatids separate and ultimately are put into different cells in telophase 2 and cytokinesis. The end result of meiosis 1 and 2 are 4 daughter cells that are haploid in number. All right. If this process were taking place in a male, this is called spermatogenesis. This takes place in the testes of a male animal, and the end result would be 4 sperm cells. In a female, it's just a little bit different. The process is called oogenesis. And what happens in oogenesis is there are 4 cells formed, but only one of them gets most of the cytoplasm. The egg cell gets most of the cytoplasm, and the other 3 cells are called polar bodies, and they're discarded. They're basically destroyed. But this is the cell that will be released during ovulation and potentially fertilized. Okay, that's meiosis in a nutshell. I hope that is understandable to you and helpful. Please go back and look at the video again if you need to hear it again. And feel free to bring your questions to class or send me a question on My Big Campus. I'd love to answer it. All right, look forward to seeing you tomorrow in class. Have a great day.